Hello everyone, this is Meghna Hada, and today I will be presenting a study on sensing and microfluidics based application of neurotransmitters to the brain. First, a little introduction. A brain is an organ made of soft tissue which functions as the coordinating center of sensation, intellectual and nervous activity. That is, it controls all physical actions, reactions, and our thoughts. For most vertebrates, like humans, the brain is located in the head inside a strong bony structure called the skull. In humans, the cerebral cortex contains approximately 15 to 33 billion neurons, each connected by synapses to several thousand other neurons. These neurons communicate with each other with a combination of electrical and chemical signals. As we have learned in class, the electrical signals are the action potentials. Neurotransmitters are the chemical messengers that carry out neurotransmission. They transfer information from one neuron to other or to other cells via a chemical synapse. As shown in the figure on the left, neurotransmitters are released from vesicles and synapses to the synaptic cleft, where they are received by receptors on target cells. Neurotransmitters help in everyday life and functions. Sometimes, the natural balance of these can be disturbed either naturally or through the consumption of drugs or supplements. The second figure on the right shows an overview of some of the neurotransmitters and the actions and intellectual impulses they are responsible for. A short summary on the entities. GABA and glutamate are the two most abundant neurotransmitters within the brain, and they work in balance with each other. GABA is inhibitory and glutamate is excitatory. Glutamate stimulates our senses and enhances learning and memory. GABA, on the other hand, calms and relaxes the brain on a psychological and physiological level. Dopamine and serotonin are two complex monoamine neurotransmitters with opposing interactions. Dopamine is the reward neurotransmitter. It is excitatory and has a large role in memory, learning, focus, motivation, and social behaviors. Dopamine is the primary neurotransmitter thought to regulate our goals and desires. Low dopamine can lead to depression and memory loss in some people. These low levels can even make us feel a lack of purpose or enjoyment due to the missing reward aspect. Serotonin has less of an impact on focus and motivation, and more of a connection with memory and learning than dopamine does. It is less of a nootropic neurotransmitter and more of a mood regulator than dopamine. Although it has been a target for many antidepressant medications like SSRIs, it generally has a stabilization effect. Acetylcholine and glutamate are the primary targets of nootropics such as paracetam and other racetams because they are believed to have fewer side effects and addictive potential than dopaminergic targets. Why sensing these neurotransmitters is important? Well, to help researchers from the medical field in their quest to unravel the human brain's neurological functioning. You do require this given the 33 billion neurons and their complex networking. It's also important to provide the critical information to better understand multiple diseases associated with neurotransmitters concentration imbalance, such as Alzheimer's, Parkinson's, schizophrenia, and Huntington disease. Sensing is important since sometimes experimental setup is more complex to handle while using live specimens having brain dysfunctions, such as the neurodegenerative disease. For example, the microfluidic design in the cortex area can be made with flexible electrode support, while deep brain research requires solid substrates. These issues are even applicable with brain slices. In this presentation, I would like to discuss 3 applications in detail. The first application is wireless instantaneous neurotransmitter concentration sensing system, or also WINCS. It measures extracellular neurotransmitter concentration in vivo, which means inside the living, and displays data graphically in almost real time. It employs two electroanalytic methods. The first is the FPA, the fixed potential amperometry, which measures neurotransmitter concentrations at an electrochemical sensor, which is basically a carbon microfiber electrode. And the second is the FSCU, the fast scan cyclic voltammetry. WINCS comprises of a battery-powered patient module, the diagram on the left, and a custom software application, the diagram on the right. The application is usually used through a personal computer. For this trial, dopamine, glutamate, and serotonin were selected as the neurotransmitting analytes. This principle was tested on pigs and rats via in vitro injections and in vivo measurements, that is within moral and ethical boundaries. For FPA, the patient module impresses upon the sensor a constant potential, but for the FSCV, we need a time-varying waveform. The resulting current is converted to a signal by the transimpedance amplifier, and it is digitized and transmitted to base using the Bluetooth. WINCSware controls the parameters for FPA and FSCV and records transmitted data stream. For the FSCV, we can see on the left that it produces 4 useful plots, out of which 2 are shown. The first is a parametric plot of raw sensor current versus time, and the second is the background subtracted current versus applied voltage. Both are displayed on scan-by-scan basis. The FPA, however, as you can see on the right, only produces a current versus time strip. FPA versus FSCV. FPA can only measure oxidation current, but FSCV, on the other hand, offers greater specificity when it comes to resolving the electroactive chemical species adjacent to the working electrode. It not only measures oxidation current, but also reduction current during the negative sweep, creating 2 distant current peaks in the resulting voltammammogram, a parametric plot of current versus applied potential. The second technique or application is the time-based CMOS potentiostat. It measures electrochemical redox currents for detection and measurement of neurotransmitters. This CMOS potentiostat is controlled by an adaptive sampling algorithm to increase power efficiency, input dynamic range, and sampling frequency. This sensor is suitable for neurochemical in vivo monitoring specifications. The diagram on this first slide shows a basic block diagram for the devices used to monitor neurotransmitter concentration in real time. Communication between neurons in the brain is dependent on chemical stimulation depending on neurotransmitter release and uptake. An integrated potentiostat performing fast electrochemical analysis is suitable as a detection circuit due to its small probe size, high sensitivity, and temporal resolution. Potentiostat electronic circuits used to measure a current generated by a redox reaction occurring between the conductive electrodes and an electroactive composition or part of interest. This can be done by maintaining a potential difference between a reference electrode and a working electrode. The transient Faradic current measured at the working electrode is directly proportional to the molecular concentration. But for constant potential amperometry, a fixed potential difference between working electrode and reference electrode is applied, allowing continuous monitoring of analytes, generating either oxidation or reduction reactions. This slide shows the potentiostat circuits used, the measurements for which rely on the charging and discharging time of capacitor. Potentiostat designs require an external digital circuit for data acquisition. This experiment uses a time-based potentiostat architecture controlled by a bidirectional digital circuit that performs a simple adaptive sampling algorithm and real-time data acquisition, which is on this slide. Various designs were compared to come up with the one with the ideal and the least power considerations. The next application discussed is the microfluidic platform for neurotransmitter sensing based on cyclic voltammetry and dry electrophoresis for in vitro experiments. A microfluidic platform that can simultaneously measure and locally modulate neurotransmitter concentration in a neuron network. While this application also uses a potentiostat, unlike the previous experiment, this one also uses an electrode functionalization to detect several neurotransmitters simultaneously. This system uses 320 bidirectional electrode arrays for dielectrophoretic manipulation and cyclic voltammetry. Each electrode is connected to a mechanical multiplexer in order to reduce noise interference and fully isolate the electrode. The same can be seen in the diagram shown. The experimental setup is very complex to handle while using live specimens having brain dysfunctions. For this, we need to understand molecular exchanges of neurotransmitters so we can successfully carry out brain mapping. Both neurotransmitting sensing and modulation must be linked together. Here, a digital microfluidic approach is used. This can modulate the neurotransmitter concentration at the synaptic gap through an electrode array and a microfluidic platform. This diagram shows the proposed platform with the 320 bidirectional electrodes. The neurotransmitter used here for analysis is dopamine. This figure shows the setup that was used. In the first part, you can see the 320 electrode array, and in the second part is the designed electronic interface of the potentiostat. This figure shows the results obtained from the experiments. Curves for dopamine solution in the range of 0.25 microM and 7.5 milliem. Figure 2 shows the results with dopamine and implemented potentiostat on oscilloscope. The given system can be expanded to a larger EA size with the full control of system parameters. There are a lot more applications of neurotransmitter sensings. An example of few are given here. Future work on the applications discussed. For Wings, the creators are focusing on improving performance and adding functionality, including electrophysiology recording and stimulation capability. They also want to create a version that facilitates the placement of deep brain stimulating electrodes or even provide feedback for the neuromodulation. For the third technique, it can be expanded, the microfluidic platform can be expanded to a larger EA size with the full control of system parameters, which will require modifications in order to support electrowetting, such as adding a Teflon layer over the electrodes and reducing the gap between the electrodes. These are some of the references that were used in this study. The end. Thank you.